Wow, what a great day so far. I'm really enjoying, I hope all of you are enjoying the symposium as much as I am, and I'm very excited here for our last and final session. And so let's get right to it. So our first speaker is Greg Thurber. He's an associate professor of chemical engineering and biomedical engineering at the University of Michigan. He's been studying the fundamental biotransport principles to design novel therapeutics and molecular imaging agents. Greg received his PhD training here at MIT. He did a postdoctoral training over at MGH and Harvard Medical School. He's authored more than 75 papers and book chapters, invited more than 120 talks at various pharmaceutical companies, national and international meetings, and universities. And he's consulted and collaborated with more than 20 different companies, including on several projects related to today's topics, ADCs, including some that have been FDA approved. Greg.
Well, thank you very much for that introduction, and really thanks to the organizers for having me here. So both my PhD advisor, as well as I saw in the audience earlier, my postdoc advisor, are here in the audience. So I guess having both my academic parents here, I will be on my best behavior. Thank you. But I really appreciate the opportunity. Oh, sorry, 1 second. Okay, it was just the microphone. I was worried they whisked me away. They didn't want to hear what I had to say. But so again, thank you for the opportunity to come here and share some of our work in the space of ADCs. It's very exciting. The field has really taken off in the past few years, and it's been a pleasure to collaborate with different academic groups as well as different companies. I'd like to start by thanking the people who made this possible. So all the graduate students working hard in the lab, a few collaborators, as well as, of course, the funding, which is essential to move the field forward. So a brief disclosure, we have worked with a lot of companies, and we've been very fortunate to work with a lot of these drugs that are going into the clinic or in the clinic, and we're being able to apply some of our concepts and delivery to molecules that are actually out in the clinic helping people. So we're very excited about that. So many of you may have followed the ADC field for some years. I won't give the full history, but as you've heard from some of the excellent earlier speakers, ADCs have been around. They even predate monoclonal antibodies. So the first immunoglobulin conjugates went into mice in the 1950s, and there was actually a clinical trial in melanoma where they did both intratumoral injections as well as systemic IV delivery of immunoglobulin drug conjugates in the 1970s. But the field really took off with the advent of monoclonal antibodies, and in the 80s and 90s, there are many different hurdles to be solved along the way, immunogenicity, delivery, et cetera. But the first approval was Mylotarg in 2000. But as you heard from earlier, there was a silent decade where that was the only drug, and it then got removed from the market. And I actually started my lab right about in this space right here. So I'm very grateful to all the people who have made this field a success, because if this would have stayed level, I'd probably be looking for another job right now and not working in ADCs. But it's very exciting to see this take off. And one of the things we're really excited about is these blue lines here. And these are the FDA approvals. There's other approvals worldwide so far, but these are the approvals in solid tumors. And that's really what I'm going to be focused on in my talk today, looking at delivery to solid tumors, which are both the more prevalent type of cancer, but also often the more difficult to treat. So we're excited that these agents are making an impact there. And as you've heard, there's been many different failures. This was not a lack of investment, but there's a lot of investment, a lot of lessons learned. And I think with a lot of new people coming in the field, it's very important, as you'll hear, as you've heard earlier from some of the people who have been working in this field for decades, to apply those lessons that were learned over those many years. Now, what does our lab look at? Well, we're primarily looking at the quantitative pharmacology of these molecules. And so there's two major challenges. It's a simple concept, but it's very difficult to apply. I'm very excited. I think there's a lot of potential for these drugs, but we need to do it with our eyes wide open. When you deliver these drugs, these molecules will bind to the tumor, but they go everywhere in the body. And so one of the main things to keep in mind is when you inject these drugs, the vast majority does not end up in the tumor. And I'm showing this here. This is not our work. This is out of Elizabeth DeVries' lab. This is a radiolabeled antibody. But with all the excellent chemistry that was done and has been done, these molecules are behaving more and more like antibodies. They're not rapidly clearing when you put all these lipophilic molecules on there. We can engineer them now where they circulate for long times. But the vast majority does not end up in the tumor. And you can see this with these radiolabeled antibodies because you have this diffuse gray haze over the entire body. And it does concentrate. So you can see some of these darker dots here. It does concentrate within the tumor. So you get a higher concentration, but still a vast majority of that dose is metabolized elsewhere in the body, releasing the payload. And it's internalized there. So no matter how stable that linker is, it's eventually going to get taken up and metabolized and released somewhere. So that's step one of the major problems. The majority doesn't reach the tumor. The second problem is what does get into the tumor is poorly distributed. And that you can see here with this image. We published this in collaboration with Evan Rosenthal, was at Stanford, now at Vanderbilt, where they were making fluorescently tagged antibodies for intraoperative imaging to make sure you have clean resection margins. But a nice part about that is when you take out the tumor, you can look where that fluorescence is. And you see this very heterogeneous fluorescence. And we've known this for a long time. We've known this since the 1980s, that these molecules are poorly distributed. And now, through studies that are done here as well as elsewhere, we know exactly why these molecules are distributing poorly. But these are two major problems. So very little gets into the tumor, and what does is poorly distributed. All right, so how do we start to design molecules that are more successful? This is a busy slide, but we've published this, so I just want to give you the take-home message from this slide. With all the success that has happened in the past few years, we did a retrospective analysis looking at the clinical success relative to the preclinical models. So there's been a lot of talk about how these molecules don't translate well from mice to humans. And I would argue that if you dose them properly, you actually do get a good correlation between the efficacy in mouse models as well as the efficacy in the clinic. One of the major problems is the payloads that are used are extremely well tolerated by mice. And if you're using a xenograft, those human tumors are extremely sensitive. And so you have this artificially large therapeutic window where the mice tolerate these huge doses and you wipe out the tumor. But when you go to the clinic, you have toxicity problems, you have to dose much lower. So what this slide is showing is we looked at it was 6 when we published this, but here are 2 new approvals in solid tumors within the past year since we published this. If you look at all these mouse models, you can find at least one tumor type that is sensitive when you dose the mice at the clinical MTD in the mice. So the exact same mg per kg. So if you give 3.6 mg per kg in human, you dose the mice at 3.6 mg per kg. And when you do that, if you look at the efficacy after a single dose within the dose window of the ADC, you see regression of the tumor. So every single approved agent, you can get regression of the tumor with a single dose when you're giving it at the clinical MTD. And so this is what we would call a clinically relevant dose. We also looked at 37 discontinued ADCs. And we looked for any example of finding a tumor that responded at the clinical dose. And only 7 of the 37 were we able to find tumor shrinkage. And some of those actually had PRs and I think a CR in the clinic, but they were withdrawn for, let's say, business reasons. So only 19% of the discontinued agents had efficacy. So the point of this argument is that if you dose the mice with a clinically relevant dose level in mg per kg, the efficacy in the mice does actually track with clinical efficacy. And so it's very important to hold back and only deliver the dose that can be tolerated in humans in order to look at this correlation. All right, why is that mechanistically? So we looked at that example, but we kind of knew ahead of time what we were looking for. So I would say 2 reasons. One, we pulled a lot of data from the literature. Most of this was from radiolabeled molecules. And we looked at the biodistribution. And so the typical units of biodistribution are percent injected dose per gram. And this is widely used with radiolabeled molecules. But I often think it's not extremely intuitive. And so what I would argue the better units to think about would be in terms of concentration. One of the problems with percent injected dose per gram is it's proportional to body weight. And so when people look at mice, in blue here, you have very high percent injected dose per gram. When you look in humans, it's very low. In fact, it's percent injected dose per kilogram that's usually used. And so there's a 3,000-fold difference between the percent injected dose per gram, but that's also the difference in body weight between mice and humans. If you change the units in terms of concentration, it's actually fairly similar, not statistically significantly different between the dose that reaches the tumor in mice and the dose that reaches the tumor in humans. So we have a similar dose level when we match the mg per kg dosing. By the way, the FDA argues for straight mg per kg dosing in terms of safety. Here we're showing it's important for efficacy as well. So we're not doing any sort of allometric scaling, trying to worry about faster clearance in this example. So that's one. While we're on it, I also graphed in green here. These are radiolabeled small molecules. Notice that it's much lower and statistically significantly lower. And this is also after, let's say, 10 minutes, right? Small molecules, classic chemo is very transient. So even the highest concentration is still below what you have in ADCs, and this concentration is maintained for days, whereas small molecule chemo rapidly clears. So this is where you get the benefit of efficacy. You have much higher concentrations on target for longer when you attach that small molecule to an antibody. All right, so that's one reason why we want to dose mice at the same mg per kg to match the concentration. But another critical aspect is the distribution. And so here we're showing one example in mice, another example from a clinical tumor that was resected from a patient, and we're showing that the tissue distribution, the penetration into the tumor, is similar between mice and humans. And so for these two reasons, both the macroscopic delivery and the microscopic delivery is similar when you match the dosing. And so I'd argue that's a critical point when you're doing preclinical testing of these molecules. All right, why is tissue penetration so important? Well, we understand what is driving tissue penetration, and one of the primary reasons are these biophysical mechanisms, that the binding rate of these molecules is much faster than diffusion. And so there's two main limitations of reaching the cell. So as soon as a cell extravasates, it binds the first target it sees, and you have to really saturate one cell layer after another, and so you have this saturation front moving through the tissue. We talked about small formats. If the format is small and it clears very quickly, let's say by renal filtration, eventually there's no more free molecule and your saturation front is frozen in time. But with monoclonal antibodies, they're some of the longest circulating molecules we know of, and so they can stick around for much longer. The problem is that for most of these, they have to bind and internalize to release their payload. And this is essentially a catalytic mechanism degrading this, and so if the rate at which the drug enters the tissue is equal to the rate at which it gets degraded, you'll never penetrate deeper in the tissue. And I don't have time to go into it in detail, but there is no peeling the onion at this scale. You cannot kill these cells fast enough to then diffuse beyond it. The kinetics, vascularization just don't work out to allow you to do that. And so we see this in animal models. So, and I showed you the clinical examples earlier, but here are some blood vessels in red, ADC in green. You can see this paravascular distribution, and it doesn't improve over time, nor does it improve with additional doses. By the time you give the second dose a week or 3 weeks later, the first one has already been degraded, and you don't reach all the cells. So this is one of the major reasons limiting efficacy. And I don't have time to go into it. It's a separate talk. But I want to point out that bystander effects are not able to overcome this. They can help. What is bystander effects? This is where the small molecule can diffuse out of the targeted cell into an adjacent cell. One of the challenges with ADCs is it's a highly quantitative science. So there's never a yes or a no answer. It's always a state of degree and a fractional efficacy. But if you're going to remember one number in your mind, I would argue 50%. So typically, if the payload is released outside of the cell, it's only about half as effective, and we've done this experimentally and computationally, only about half as effective as if it's internalized into that cell. Think of a boulder, and you have a bunch of explosives, right? If you want to remove that boulder from the middle of the room, you can either put a whole bunch of explosives next to the boulder, or you can drill a hole into the boulder, put your explosives, and set it off, right? That's much more effective. So this is one of the reasons why you might see some efficacy, and people are talking about other mechanisms like Fc gamma mediated uptake and release by macrophages, or extracellular protease release. That can all be beneficial. However, it's not going to be as effective as targeted therapy. So I would argue it's still important to engage that target to drive very high efficacy. All right, so I'm arguing that this tissue penetration is very important. How do we improve the efficacy? And there's a long story behind this. I'll just kind of walk through a few stages in our lab that we looked at. One of them was looking at the drug to antibody ratio. So this is one of the first experiments we did. I was assistant faculty member working on a review paper, and we wanted to compare different drug to antibody ratios. Now it's important to keep in mind that the payload is what causes the toxicity in almost all cases. And so you're limited by the payload dose. And so what we showed was, when we did this retrospective analysis from the literature, in every example where there were fewer payloads per antibody, a lower DAR, but they gave the same total, same total payload dose, you had better efficacy. Why? Well, you had to spread out those payloads over more antibodies. That higher antibody dose was able to penetrate farther, and you saw a stronger response. There was only one counterexample, and that was in the case of what became Trodelvy, where they had a very low potency payload, and they were giving very high doses. They saturated the tumor. And so when you saturate the tumor, there you need to have a higher DAR, try to deliver as much payload you can into each cell. So the main result from this slide is that a lower DAR can often be beneficial. It's less potent, but it allows you to penetrate deeper in the tissue. Now, we submitted that and was reviewed, it was accepted, but one of the arguments, and a fair argument, is that with lower DAR, this was earlier in the field where the chemistry wasn't perfected. We didn't have all the fantastic chemistry that you heard about earlier, and when you have more drugs per antibody, that can speed up plasma clearance. We analyzed this, showing that that wasn't a major driver, but it was still a retrospective study, so we couldn't completely rule that out. So we wanted to come up with an experiment. How can we show it's really the tissue penetration and not some sort of faster plasma clearance by changing the DAR? So we wanted to keep the DAR the same, but change the antibody dose. So the simplest way to do that is just spike in more antibody. So we took Kadcyla, trastuzumab with DM1 payload. It was the only approved ADC in solid tumors at the time, and we spiked in different ratios of antibody to partially block some of these binding sites. And when we do that, we get better tissue penetration outside of the blood vessels. It's the same ADC dose, same delivery of the ADC to the tumor, but now we're spreading it out more evenly. And this was published many years ago, but we chose a model that was not sensitive to the antibody by itself. So just the antibody in this model doesn't have a benefit. If you did have some type of antibody benefit, this would be additional benefit for the patient, but in this model, we ruled that out. The ADC by itself has some efficacy, but when you spike in 1-to-1, 3-to-1, or 8-to-1 levels of antibody, you see significant improvement. This was very counterintuitive at the time because everyone was trying to make sure you had a payload on every single antibody, but we were showing that if you do this in a petri dish where there are no delivery limitations, it hurts efficacy. There's no efficacy in and of itself, but if you do it in vivo, you see better efficacy. It's because it allows better tissue penetration. This started several collaborations with different companies. One of them was with a company called Immunomedics. And they had a molecule where they had put SN38. This eventually became Trodelvy. But they used a much lower potency payload. So I've shown you two ways to solve tissue penetration. One is lower the DAR. The second one is to add antibody. A third way is to use a lower potency payload. And I would argue this is one of the big drivers allowing you to dose higher, and one of the reasons behind some of the success of the topo-1 inhibitors. You can dose them higher. Even though they're higher DAR, they're still less potent than microtubule inhibitors. So we showed that TROP2 is very rapidly internalizing, and you have to dose it at fairly high levels in order to penetrate into the tissue. But with Trodelvy, the lower potency payload, you can do that and reach all the cells in the tumor. I'm not showing it here. We published this a couple years ago, but interestingly enough, this molecule is dosed on day 1 and day 8 of a clinical cycle. By day 8, you still have some of the circulating antibody. The other important thing I should have mentioned was this payload falls off very rapidly from the antibody itself, very unorthodox ADC. So by day 8, you mostly have antibody circulating around, and it actually acts as a penetration enhancer because it allows the dose of ADC to penetrate deeper in the tissue. And so we showed that in animal models as well. I won't steal the thunder of one of the other speakers, but there are also examples where the lower linker stability can help with tolerability. And so with this molecule, because that payload falls off, you don't have a lot of target-mediated toxicity. I don't have time to get into the details, but one of the take-home messages for ADCs is you're always trying to find a balance. You don't want to be too extreme in any direction, or you'll end up maybe causing new problems, even though you solved one. All right, we also had a collaboration with Immunogen. And so this is a molecule that ended up becoming Elahere. So this is mirvetuximab. We weren't allowed to call it that at the time. It wasn't approved. But this is a great collaboration. They had a lot of excellent tools at this company at their disposal. And this is a anti-folate receptor alpha molecule with DM4. So I talked about payload potency. Here's another example. With this DM4 payload, it's very potent, but it's not as potent as this DNA alkylating molecule. So this highly potent DNA alkylating molecule is not well tolerated, so you have to give it at a fairly low dose. So if you have a high expression tumor, by using a lower potency payload like DM4, you can dose higher. In this case, we dosed at 2.5 mg per kg, and that allows you to penetrate deeper in the tumor, and you can get complete responses in the mice. In that same tumor model, if you use this high potency payload, you have to dose lower, less than a mg per kg. You don't penetrate very far, and now you see a lower response. So this is another area of balance. You need to balance the potency of the payload and the DAR and the dosing in order to tailor it to a specific target. There's no one size that fits all. And as an example of that, when we took those same molecules and we went into a tumor model that was much lower expression, this is an OV90 tumor model, and now you have the opposite case, where you can dose very high, 3 mg per kg, you're saturating the tumor, but there aren't enough binding sites in order to kill these cells. And so in this case, you get some response, but not complete killing with the lower potency payload, but this higher potency payload, there's fewer binding sites, you penetrate farther, even at a very low dose, and now you do have the potency. So it's really important to balance all these factors for your particular target in order to tailor that efficacy. All right, how do we predict the optimal design for a given target? And I know I'm going to run out of time, so I won't spend a lot of time on this. There was an excellent talk earlier this morning by Alison Betts on this. But in our lab, we're using some agent-based modeling. This is in collaboration with Jennifer Linderman, and actually one of my former students is, I think, in the audience here, who did this work. But essentially, we're overlaying these partial differential equations of looking at drug distribution and bystander diffusion in the tissue. We overlay that on a grid of thousands of cells, and each one of those cells can divide or die depending on the local concentration. We can account for systemic distribution, and we can also account for the cellular and subcellular distribution. Now, this model took many, many years, so Ishta spent a lot of time on this, and it was grounded in work that I had done in both my grad work and postdoc. So it's very complicated. We're going to go through each one of these parameters one at a time now. But no, so all of this has been directly tied to measurements that have been done in vivo. And of course, we took a lot of data from the literature in order to properly train this. But we can now make predictions and actually do quite well at predicting what the efficacy is going to be in the mouse models. And what the nice part about this agent-based model is we can directly compare the simulations and looking at the perivascular distribution with different antibody doses with the histology, taking out tumors, slicing them, and looking where the drugs go. And it also predicts efficacy very well. More recently, we've been adding immune cells and training the model to incorporate the immune effects to improve the predictability of these simulations. But of course, we're engineers, and so we want to do the detailed simulations, but we also like to boil it down to some fundamental parameters. And I would argue that the Thiele modulus, which is a chemical engineering principle, originally developed for the diffusion of reactants into a catalyst pellet, so you didn't waste your money on expensive catalyst in the middle if you didn't need it. The same principles apply excuse me, to ADCs. And in fact, when I was joining Dane's lab, I think he was actually the one who originally derived this for an antibody and said, see if this works with antibodies. And it did very well, and it continues to do very well in predicting efficacy. Why do I say that? Well, we took all the approved ADCs at the time that this was published, and we looked at this dimensionless number. All you need to know is it's a ratio of supply and demand. The demand, or the internalization, is the internalization rate times the target expression. And the supply is the permeability of blood vessels and density, and your antibody or ADC dose. And if this ratio is close to 1, you're dosing at a level to reach most cells in the tumor. And in fact, what we showed was all the approved ADCs in solid tumors were dosed where you were balancing these factors. And so what this means is to be a successful ADC, you need to have a dose high enough to reach all the cells in the tumor. And that's not always the case for other drugs. These are checkpoint inhibitors where you don't have the toxicity. You can dose these extremely high. And so now instead of a ratio of 1, you're down to 0.1 or 0.01. You're super saturating the tumor, which you wouldn't be able to do with an ADC. All right, I only have time for one concept left, so I'll skip the last one. But I'll show you an example of the fact that we can predict the distribution of these molecules. How do we solve this expression problem, right? So you could take a patient with high expression and co-dose an antibody and a patient with low expression and not dose, but many patients have different metastases that have heterogeneous expression. So how do we tailor the delivery in order to maximize efficacy? And so the ideal scenario, if you have high expression and you just have your ADC that's poorly distributed and you have low efficacy. So if you add in this extra antibody, you get better penetration and better efficacy. The problem is in low expression, if you do the same thing, now you block too many binding sites and you lose your efficacy. So ideally, we'd have a scenario where this green antibody competes for binding with high expression, but not with low expression. And if it did this intrinsically, it would automatically tune the delivery of the payload in the cells. And so to make the long story short, in collaboration with Peter Tessier's lab, we engineered these molecules. We used an avidity effect, where if we reduce the affinity of one of these arms enough, with high expression, both arms can reach and bind easily, so it can compete. But with low expression, both arms can't bind very well, and so the higher affinity antibody outcompetes. And so it allows us to do this auto-tuning. And so here is this, we call it a high avidity, low affinity carrier. We get better tissue penetration. I'll just jump to the results here. In a high expression model, when we have untreated tumors and we combine them with just treat with Enhertu in blue, we get a good response. Enhertu is a great drug. But when we add in trastuzumab to Enhertu or our HALA antibody in red, we get better tissue penetration and better efficacy because the direct internalization is better than the bystander effect from that molecule. But interestingly, when we go into a low expression model, we get good efficacy with Enhertu. Again, it's the DAR8 molecule. It's pretty potent. When we add in trastuzumab, though, we block binding and uptake, and so we see very poor efficacy. But with our HALA antibody, it's not able to compete on the low expression level, and so we maintain efficacy in that system. So this is a way of co-dosing an antibody that automatically tunes the binding in order to optimize the delivery. And it could happen even within the same patient. It could be 2 different tumors, 2 different metastases, because it happens automatically. There's a lot of fun facts about this that I don't have time to go into. We engineered the Fc domain. So in low expression tumors, normally you would only have the ADC binding. But we enhanced the Fc domain, and so if there's an immune cell around, the hands are too weak to hold on, but once you bind to the Fc domain, within that immunological synapse between the cancer cell and the natural killer cell, it can outcompete the ADC and drive strong Fc effector function. So we're trying to maximize multiple mechanisms of reaction, multiple mechanisms of action. All right, I'm over time. I won't go into combination therapy, but I just want to summarize with a couple take-home points. One, we need to use clinically relevant dosing in animal models. That's important in order to ensure that we have molecules that are going to be successful in the clinic. We can use multiple approaches to improve tissue penetration, such as optimizing the DAR, lower potency payloads, or co-dosing, or smaller formats. And ideally, we can combine computational models with our experimental work in order to more efficiently design molecules that are going to be highly effective in the clinic. So with that, I think I'm out of time. I'll end there, and thank you for your attention.
Okay, a couple questions. Go with that side first.
Okay, sure. Okay.
Hi, thank you for that. I'm wondering, as you add in these trastuzumab decoys, I guess you could consider them as, on one hand, there's the blocking of the binding site. I wonder what happens to the PK and the circulation parameters as you increase the dose of a blank antibody.
Right. Actually, there's a lot of additional benefits that I didn't have time to go into. So in that immunogen paper, they also generated a cross-reactive antibody. In healthy tissue, it's much easier to bind your target. And so when you add in that antibody, you can completely saturate the healthy tissue. So there's analogies to this in the radioimmunotherapy field with Bexar and Zevalin. You cold dose the antibody to block healthy tissue. So by increasing your antibody dose, you're lowering the uptake. And we showed this in the mouse model. You lower uptake in healthy tissue. You circulate longer because you don't have that target-mediated drug disposition. So you have better circulation, and so you get more drug in the tumor. So there's multiple benefits. Less uptake in healthy tissue, more total drug in the tumor, and better tissue penetration in the tumor. So all these factors, I think, contribute to the improved therapeutic window when you optimize those.
So should this be a sequential dosing where you give the trastuzumab first, and then you give?
It depends on the details. The short answer is oftentimes it doesn't help as much as you think because with the 7-hour half-life or 6.5-hour internalization half-life, you quickly re-equilibrate. So if you gave the cold dose and then the hot dose, it doesn't help you as much as you think because you re-equilibrate as soon as you give the ADC. But there are cases, I think Andy Boswell at Genentech published a nice paper where you downregulated the healthy tissue. And so that's much longer lasting. So their cold dosing and then coming in can be beneficial. So it depends on the specific target, I would say, in terms of the benefit.
Thank you.
One more.
Yeah, thank you. Have you compared stochastic versus site-specific conjugation for tissue penetration? So for the background, stochastic is more heterogeneous, site-specific is homogeneous distribution. Yeah.
Right, so I would say it depends on the biophysical properties. If you can do stochastic labeling and you still have good biophysical properties, or let's say semi-site specific, right? Hinge region would be semi-site specific. If you can maintain your biophysical properties, I think the tissue penetration is primarily driven by the number of binding sites. But there are some advantages to the site-specific. I mean, obvious manufacturing advantages. You can avoid any sort of binding with or interference with Fab or FC domains, but also the improved biophysical properties. So I would say that's probably what I would keep as the most important. Make sure you have good biophysical properties of your molecule with the chemistry.
Thank you.
Thank you.
Great. Thanks, Greg.